we were lucky enough to snatch him and bring him to the FNNDSC in the early years. For a number of years, we worked really closely with Rudolph developing the CRISP infrastructure, which we've continued to rely on and use, daily, among the team. And we're starting to get more and more interest in it around, other research groups as well too. But Daniel was really key in the beginning days of getting that whole platform up and going. And from there, we're really proud to see him go off to do a PhD at Harvard. And I was lucky enough to actually go with him to interview with the person he wanted to work with there. And we were just so excited that he got accepted into the Harvard PhD program and in computer science. And after graduating there, he's now an assistant professor of computer science at the University of Massachusetts, Boston, and an associate of Harvard Paulson School of Engineering and Applied Science. One theme that Dan and Rudolph and I have all kind of centered around and really embraced is this open science and open education. And that's something that Daniel was really pushing forward with our group in the beginning years as well too. So it's really exciting to have him back. So now he's going to give us a talk on brain connectivity, machine perception, and computer graphics, all at different scales. So Daniel, welcome. It's just so exciting to have you, and I'm looking forward to hearing what you're up to.
Thank you very much, Ellen. So in my bio, I said working at the FNNDSC was one of my best chapters in my professional career, and that's true. I don't know if you remember at the goodbye conversation we had. I said I made real friends in this group. I think you have a fantastic crew, and I'm sure it's still the same. Very nice and talented people. So I enjoyed my PhD very much, and I love my current job. But the time at the FNNDSC, really, I will never forget. And with that, I will share my screen now. I hope this works. Let's see. So can you see that? I shared a browser window.
Yep, I see your browser window.
All right.
There's no, it's blank right now.
Yes.
Okay.
All right. So I was in the FNNDSC 2012, 2013, and one of the things we did back in the day was develop XDK, this first WebGL toolkit for medical imaging, and then this application SliceDrop. So I'm sure some of you know and remember that, but I want to quickly go back to 2013 to this video we made to present SliceDrop at SIGGRAPH. And SIGGRAPH was, if you're not a computer scientist, maybe you don't know, but SIGGRAPH is for computer graphics, the top conference. So actually, if you get one paper at SIGGRAPH, you can probably get a PhD at Harvard. So let's quickly look at this video. It's from 2013, very amateur video we submitted back then. I just want to show you. Hopefully this goes well here. Let's see. So it's SliceDrop, this web-based video we can drop in the medical imaging. Oh, I don't know why.
Oftentimes such software exists in medical workstations. These packages are complex to use, large, and overloaded.
We present SliceDrop, a
That's me.
video for medical imaging.
You saw Rudolph.
2D and 3D using WebGL. Features real-time rendering and real-time sharing across devices.
Go to SliceDrop.com to visualize your data, share it, and collaborate in one click. It's free and open source.
That's right. So let's move a little bit ahead. So we show SliceDrop, and then I want to move here, I think. Let's see. Yeah, so
all scene and camera information.
Maybe a little further here.
This, we believe, will greatly simplify and enable real-time collaboration in medical research and medical imaging.
Oh my God, I love SliceDrop!
So yeah, I wanted to show this. I thought we had so much fun doing this video, and then we presented it in front of I think it was 1,000 or 2,000 people, the SliceDrop application, and it was a live presentation with the Wi-Fi, and everything went well, and we were so proud. I remember that. So I hope Rudolph, you remember that as well. I don't know if Nicola is still involved, but yeah, he was there as well. So good times. So then in 2013, I left the FNNDSC to go for my PhD to Harvard. And 2019, last year, it was, I finished, you know. So here is a picture of the graduation, and you might already notice that all the people wear those little tiny black hats, but I have like this huge thing on my head. And what that was a special hat my lab mates made for me, which looked like this. And then somehow through this hat, people looked at me, the people approached me, like a journalist took photos, and that was pretty fun. It made me feel very special during that day. And you can actually buy the picture of this hat taken from the Boston Globe for hundreds of dollars, just because they thought it was so cool. So I really appreciated that my lab mates did that, and it was fun to wear. It was very heavy, so I wobbled around a little bit, but yeah, still a lot of fun. All right, so I graduated with my PhD last year, and then I directly joined the faculty here at UMass Boston on the tenure track, and my research group here is called Machine Psychology. And machine psychology is a very like bold term, you know. People ask me all the time, oh, what does that mean, you know? And actually, I think I came up with it. And then now, if you go to Google Scholar and look for machine psychology, you see other people adopting this term. So I'm very proud of that. And I'll tell a little bit more what we mean by machine psychology. But first, UMass Boston, before I joined here, I did not know much about it, but it's actually a very, very beautiful campus right at the ocean, a little bit south of Boston. So it's a JFK UMass T-stop. Here's the campus, and it's surrounded fully by water. You have actually this harbor walk around the campus where you can walk. Here's my office where I am right now with some of my students. And what I also didn't know was that Barack Obama actually has an honorary PhD from UMass Boston, and so has Steve Tyler from Aerosmith. So UMass Boston, a great place to study, you know, this aside. All right, so actually I will also open the chat if somebody has any comments or questions, okay? So I said my research group is called Machine Psychology, and this is already a bold name, but I also have this even bolder claim where I say it is now time to study machines the same way we study humans. And with machines, I mean artificial neural networks. And with humans, I mean natural neural networks. So we can actually summarize that in the machine psychology research group, we like neural networks a lot, all kinds of neural networks. And as you are aware, there is still this gap between artificial neural networks and natural neural networks. And I always like to show this video by Lex Friedman, who's this deep learning researcher at MIT and very popular in the community due to the AI podcast, and very nice guy. And he allowed me to share this video whenever I present. So here on top, we have Messi doing this incredible run across the field against Getafe, like playing against all, like dribbling among all the players and then scoring. So incredible. And then in the bottom, you have a state-of-the-art soccer robot who seems to struggle to stand still even, you know. But the robot will attempt to shoot in a little bit right now, and so close, you know. But here you see the human does that way better. Of course, this comparison is not very fair. Messi is one of the best players in the world, and with the robots, we're still at the beginning. And the truth is there are cases where machines are superior to humans, or like better than humans. For instance, in chess, you know, 1996, Garry Kasparov loses against the Deep Blue chess computer. Then 2017, we have AlphaGo, an artificial neural network that beats a professional Go player. And then last year, we have AlphaStar, where the machine beats a professional StarCraft player. And this is increasing complexity from chess to Go to StarCraft. This gets more complex and more parameters to master the game. So these are examples where the machine actually beat the human. But I also like to show this one a lot. It's like recent work presented last year at CVPR, where there's a picture with a school bus, you know, and here the machine says 100% to the school bus. But if you rotate the school bus in this picture, like here or like this or like this, then the machine fails to detect it's a school bus. It says garbage truck, punching bag, snow plow, which all like kind of makes sense if you think, okay, from the orientation and so on. But us humans have no problem to say, okay, there's a school bus in all of these pictures, right? That's very, very clear. At the same time, I would like to highlight this. There was work we presented at the visualization conference in 2018, where we study how artificial neural networks parse charts and graphs. And I'll talk a little bit more about this later. But here, the method, the algorithm can measure the length of this line, no problem. You know, we trained it, no problem. But then, if you make the line thicker, you know, just this little change, the algorithm is not able to detect the length of this line anymore. And while we humans have no problems, right? We know how to measure this line on the left, we go over to the right and measure this line as well. So these introductions lead to this gap between artificial and natural intelligence. And I think, and many others think, that in order to make the artificial intelligence more robust, we need to better understand the brain. And of course, we can do that at the macro scale. We can look at brain connectivity at the macro scale with a MRI or functional MRI, diffusion MRI, right? You get like, you can get connectivity graphs, area level connectivity graphs. Okay, this is actually my brain, which Boyan scanned back in the day. You know? All right, so this is a macro scale brain connectivity. During my PhD, I focus on connectomics, and connectomics goes to the micro scale brain connectivity, and we have like some incredible data sets over there. So what we do is we take mice or rats or humans now even, cut out their brains or pieces of it, and then slice the brain up, the tissue real thin with a diamond knife. And then we scan these images with an electron microscope. And this guy here on the right is Jeff Lichtman. He's a very famous neuroscientist. He was on my PhD committee. And once you scan the data like this, you get 2D slices. I hope the video comes across. It was the zooming in, Bobby, the neuroscientist or biologist, held the sample and we zoomed in, and then we get images like this where we see actually individual cells and intracellular structures such as mitochondria here, like those dark spots, or vesicles, those little circles here for the synaptic connections. And this data can be very large because each pixel can be at a resolution of 4 nanometers. You know, so these data sets can be terabytes, petabytes of data. So very, very, very large. And what we are mainly interested in is identifying, or like the first step is identifying the cell boundaries. So which are now here, these green lines, you know, because this is the first step to generate this connectivity graph. Once we have done the synapses, we can create this whole graph structure. So this is one slice, right? And of course, we take a block of tissue, slice it up in slices, and scan them all in 2D, and then there will be a line, so we get this volume. And here's another, and then we use machine learning to identify each of these slices. I'll talk a little bit more about this. So we identify each of these cells, but what you can see here in this video is the complexity of the neuronal structures at this level. So this was manually annotated by 2 neuroscientists over the course of 6 months. It's a very tiny piece of tissue, and you see the variety of the neuronal structures. Here actually is a human hair on top, and this area, what we saw just in the video, this little, we call it the cylinders, 3 cylinders, was published in Cell 2015. Here is just this tiny red tissue, and this one are 50 microns. You know? And now we have data sets of while I was doing the PhD, we did 100 microns, which I'll show in a minute. And we also did, now we do a millimeter cube, you know, which is 2 petabytes of data. All right, so here's actually the 100 microns, and these are the 20 longest neurons automatically reconstructed in this 100 micron block of tissue. This is now a video, but it's actually WebGL rendered presentation. So it's all challenging on all levels from the computer science standpoint and scalability and so on to render these large data sets. And we use like MIP maps to show resampled data and so on. So let's take a look again at these 2D slices. So they are now aligned, you know, so these are just 3 example slices. And as I said before, these colors should identify each individual neuron. But if we look closely here and zoom in and just focus on these, the arrows show segmentation failures. You know? So because this should really look like that. There are only 2 different cells here. So the purple one and the blue one, and you see the yellow shows this falsely merged, this region is falsely merged, and this here is false, the red arrows show false splits in the segmentation. And this segmentation should really look like that, you know, instead of this, look like that. So how do we get from this? I'll show in a minute, but here, this is one big mistake of the segmentation in this 100 micron block of tissue. And I know we are recorded, but I think I can say that we lost a lot of funding because of this mistake. You know, we called it the crazy hairball. It was impossible to fix manually. So the algorithm just merged everything together. I think it was like 20 to 30% of the whole volume was merged together. It was a big, big failure of the automatic segmentation. And yeah, we lost a lot of funding for that. So, okay, so how can we fix these errors in the segmentation? And one way to do it is fully interactive. And in 2014, I published this software called Dojo. It's interactive proofreading tool, web-based for multi-users. People can look at and fix the volume at the same time, the segmentation. You have a 3D and 2D view. You can draw something and it like does some semi-automatic error correction. And people liked it. We did a user study and we compared this Dojo software, our software, against 2 existing tools. And this is variation of information. It's an error metric, so the lower the better. And we compared it against manually annotated ground truths. And Dojo was the only software where complete novices, people from the street, we recruited people from all walks of life, were able to fix segmentation errors. So this error actually went down here in the meanwhile. The other softwares, there were more for, let's say, professionals, a lot of parameters. Dojo has no parameters, easy to use, very simple, slick user interface. So people were able to fix that better. Also, the 3D view helped to orient the users. But what we noticed during these studies is that finding the errors takes the majority of the time, you know? So just looking through the volume, seeing, okay, where's something wrong, you know? Where can we fix it, takes the majority of time. And that's why we developed a system called guided proofreading. And guided proofreading looks like this. We show the user just 2 different options, and then the user selects left or right, you know. So you can click through it. Here, for instance, here you see there's a boundary missing, so the user will hopefully click on the right and accept this correction. You see the error metric here is going down, you know. So these fixes allow the user to reduce the segmentation errors. And to enable to be able to have this working, you know, to find and suggest the errors to the user, we use an artificial neural network. And it's very traditional classifier. Not many layers. What we do is like some dropout regularization, and then the network gets 4 patches or like 4 channels as an input, and spits out whether an intersection between two neurons is an error or is correct. If it's an error, it needs to be merged. So that's a split error detection. So split error looks like this, so here, and it should really look like this based on the gray scale image. So you see like the algorithm puts 3 different colors here, which means 3 different individual structures, but it should really be only one. So how this network works is we feed in 75 by 75 pixel image patches with these informations. And then at the center point, the network looks at the membrane at the center point and then says, okay, it's a split error or not. If it's a split error, merge it if the user accepts it. So we can sort by the split error score. All right, so these are the split errors. But as I said before, we also have those merge errors, and merge errors means that the algorithm merged cells together and they shouldn't be merged together. So here, this structure should really be here should be a boundary, right, between the cells. So what do we do in this case? We do the following. We first generate a ton of artificial, so we take this segment, we generate a ton of artificial borders using the watershed algorithm and some tricks with dilating the label first. That's why you have like this outline a little bit expanded. And then we take each of these generated borders and feed it to the same network what I showed on the slide before, which says, okay, this border is error or correct. And when this border, when this classifier, our algorithm says it's the correct border with the highest probability, then we say, okay, that's where we need to have the border. So we essentially invert the split error score. So that's always a little bit tricky to grasp at the first try. So for the merge errors, we have these regions. We generate artificial boundaries or borders in these regions and test each one. And if the network says this one is very correct, then we say, okay, it should be there. And that's how we deal with these merge errors. So does anybody understand that?
So when you're doing this, Daniel, do you have to, how many correct answers do you have to train it on first to get it to do that? Like you've got to train it first, right?
Yes, so we train it on, so we do the usual, which is like the 60-20-20 split, of the data sets we have, 60 for training, 20 for validation, 20 for testing. So we did take these 3 cylinders I showed in this video, which were manually annotated as the ground truth. And then we took the automatic segmentation and did the max overlap with this ground truth to essentially train the network. Then we have both, like the automatic segmentation and know where the errors are, and the ground truth manually annotated where we know that this is correct. So yeah, if you have a data set, you need to train it, you need some manual annotations, and then 60% of these manual annotations you would use. So how many patches did we use in total? I can probably quickly look that up. But the thing why this got into CVPR, which is one of the, or like maybe the best computer vision conference, is this trick, I believe, that we can deal with the same network, merge errors and split errors. And that's something I was very proud of. And let's see. Well, so all these papers are on my website too. I'll give the link later. So if you want to know more information, let's take a look. So, okay, so we put in actually a significant amount of number. We sample 112,000 correct and 112,000 error patches with the 4 channels. And then we take this data and split it. So 60% of the 112, so roughly like 60,000 patches for training. So 6,000 samples for training. So it's a significant amount of training data, of course.
Great.
Yes, but what we discovered is that using this yes/no selection for the user, it works much better. And we did the following experiment. So Dojo was the fully interactive software where the users had to look for the errors and then correct them, while there are two versions here: focus proofreading, which was an existing method, and guided proofreading, our method. And the guided proofreading just same error metric, lower is better, just was gave better results, yielded better results than the interactive proofreading method, and also the focus proofreading. So, and then what we also noticed in another user study where we compared novices to experts, and here you see also arrow metric, lower the better on the left, and then on the bottom, it's the number of corrections. And you see with the guided proofreading here in orange, the user clicks yes, no, goes through the corrections, the error reduces very fast. And in fact, for novices, it reduces at the similar slope as for the experts. And at least to a certain degree, up to here, let's say that's roughly 200, 300 corrections, where then the users get tired, So the user study were like 30 minutes, after a while the users get tired and they made mistakes. And also what you see is that the classifier at one point doesn't work that well anymore. I'll speak about this a little bit later. But the takeaway we had first, that it's possible for novices, no experience in this kind of data or proofreading or connectomics at all, were able to do stuff at the level of experts. And this is so important because the data sizes are so large that we need to have so many people help with the corrections. All right, yeah, that's guided proofreading. We followed up with a paper last year where we extended this guided proofreading to 3D based on the skeletons of the neurons. And this worked really well. We have like this, it's called like biologically driven error correction, where we have like a little look around algorithm. So we check if the branches go in a certain angle, which is not allowed, have some constraints going. And this also works very well automatically to a certain degree. And then presented this year at ECCV is another proofreading approach where we show the user not only one choice, yes or no, but multiple ones, clusters of the errors, and especially here for synapses, those are synaptic masks, and let the user accept multiple corrections at the same time by grouping them, and that works even better. So the guided proofreading already yielded like a 7-time increase in performance and throughput compared to the interactive method, and this is even much, much better. All right, so this is cell identification and error correction of automatic cell boundary segmentations. What I'm also interested in is in mitochondria. Since this data is of such high resolution, we can look at stuff. We can also detect certain other features like these mitochondria, and actually from for the human eye, and also for the algorithm, this is an easier problem, I would say, identifying these dark circles here. Even so, sometimes the algorithm does mistakes, but it's an easier problem to do. And I'm very, very interested in doing that because I want to use the mitochondria for alignment. So once we scan the data, the slices are all over the place, but we need to align them properly to this to the 3D volume. And we so far do that with very traditional computer vision features. And I think if we use mitochondria or like biological constraints, then we can improve this process. So this is what we're working on right now. I have 2 students, Yahia and Jana, who just do excellent work and looking into this. We're trying to use this ISDN and voxel morph as well, which you might be familiar with, recent published work, right? And we think we can do that at the speed of the acquisition, or the acquisition speed of the microscope, because we are able to detect those mitochondria very, very fast. And we just presented this work fast mitochondria detection for connectomics at the medical imaging with deep learning conference. And what we do is we use a unit, and we significantly reduce the number of parameters from 20 million to just over 1 million. So reduction of 94% or 93% for this unit, which it makes it so fast. So this way we can detect the mitochondria while the slices of the microscope come in. And this is important, that there's no computational backlog, you know. So we want to use these mitochondria now for alignment, and hopefully this will all work in real time. Yeah, I put in the GitHub links. I didn't mention that also for the other previous projects. So everything is open source available. So now this was the microscale brain connectivity projects I wanted to speak about, present. And now let's go to the macro scale. That's something I think is maybe more relevant to, at least was back in the day, more relevant to the FNNDSC research. So we are involved in this ABCD project with our collaborators at Brigham and Women's Hospital, which you know very well, as Yogesh Rathi and Lauren O'Donnell, Steve Piper, of course, you know. And this ABCD study, and you might be involved too, I'm not sure. Like they look at many different children with many different scans, you know, and they create the diffusion MRIs for Draktography over multiple time steps. So it's like a very long-term study of brain development, including all kind of co-founding factors like water quality, how often they play video games, and so on. So very interesting data set. So my student, Lorraine, works on this software called FiberStars. And FiberStars allows you to compare the fiber bundles, Draktography bundles, of different patients in parallel. You know, so here you have, it's a web-based software. You select the subject you're interested in, you select the cluster you're interested in, and then you have the 3D view. It's all interactive, where you can see the fiber bundles, and then you have these little fiber stars, which actually show in a 2D version, this spider chart, the mapped quantities along these fiber tracks. And with that software, you're able to explore large cohorts of patients. These are all individual patients. They are grouped together through different clustering algorithms. And then you can, like, it's called like brushing. You can brush over some, then compare them in the 3D version, but also directly look at those fiber stars to see if you have outliers in the data. So here, each row shows a different cluster bundle, and you see that some of these look more like each other, while sometimes they are, let's say, outliers a little bit. Maybe it's not so clear here, but yeah, it's faster to look at it like this in an overview view than looking at loading each file individually and so on. So also this work is available online, and unfortunately the paper just got rejected, but we're working on a resubmission right now. And I know some of you, and thank you very much for that, were involved in the user study, in the expert version of the user study. As part of this project, the FiberStars, I was personally looking into a compression for Draktography files, and not sure if you are familiar with this. It's called the GLTF file format. It's a graphics layer transmission format. It's pretty new, maybe in the last few years, but it's all the hype right now in the especially web-based computer graphics community. I call it in my computer graphics class, I call it the royal file format, you know. And the reason why I call it like this is because it's all like a hierarchy of the scene. So you can have multiple scenes in one file to view stuff, all kinds of information, volume, meshes, fibers, anything you want. It's a JSON-based format where you have the data directly available as array buffer and element array buffer. And that means without processing, you can push it to the GPU for rendering. That makes it very, very fast. Once you have this file, you can just render it on the GPU for visualization or for even real-time processing. And in connection with this GLTF file format, Google released this Draco 3D data compression. So we played around with this algorithm, and it was a little bit complicated, but we found out a way how we can use this algorithm for any kind of point cloud, not only for meshes. So this was designed for for mesh structures with triangulated meshes. So we played around, and the algorithm already supported it, but we had to create a special Python bridge and so on to allow that we can use this for Draktography data. And then we made this software called Draco. And Draco is essentially Draco for Draktography data. So what you can do is you can go and I think Rudolph will appreciate it. You can type pip install Draco, download this package, and then you have the Dracofy software, which takes in VTP, or you can also use it with TRK files, and then it spits out those TKO files. And if we look at the results, those TKO files are, it's let's say on average like a third smaller than the VTP files. And this is with a lot of added scalars and per vertex, per fiber attached values, which I know that the researchers like to do. Compression factors around 3 for standard Draco parameters. And if you have like a whole brain Draktography file, which can be from my collaborators at Brigham Women's, like 3 gigabytes, this makes a big difference. You store it as 1 gigabyte or less. So this is also publicly available, open source, and actually we just got accepted to Mikai, and we presented in October there. All right, so far so good. So my plan for today was to load off so many different projects, not go into detail, but more like the broad range of things. And if you have further questions, always feel free to interrupt me right now or reach out later on, right? So let's switch over from what I mentioned before, this gap between the natural and artificial intelligence. So now let's switch over from the brain connectivity to studies of current machine learning algorithms. And what I like to do, what I'm very interested in, is something called machine graphical perception. And this is based on a work which was done by the researchers Cleveland and McGill in 1984. And what they did is they studied human perception. And this is perception of visualizations of graphs, of charts, bar charts, pie charts, and so on. And what they asked humans to do to identify certain things in these elementary perceptual tasks you need to do. So for instance, they said, okay, if you read a bar chart, you need to understand the length measurement, right? To know how the bar D here is longer than all the other ones and so on. For a pie chart, it's more like the angle and maybe the area measurements which come into play. So these 9 elementary perceptual tasks were identified by Cleveland and McGill in 1984 and then measured how humans perceive each individual one. They rank them as well. And then they do tests like how this works with pie charts and bar charts and how humans can compare the different quantities that that are encoded visually encoded, right? So what we did is we took these 9 stimuli from Cleveland and McGill and just generated many of those artificially. We did this here for these, and for all 9, we did that for all 9, and we also did that for the pie charts and bar charts and some other experiments they presented for human perception. And what we wanted to know is how are machines doing it, compared to the humans. And for this, we pitched 4 different neural networks. And we have here so all of them end in this multi-layer perceptron as the top layers, so to make it equal. And then we compare different convolutional feature encoders, Linnet, VGG19 acceptance, standard networks, not the newest even, Linnet was first used for these digit recognition for postal codes, right? from the US Post Office. And so there's not very deep, only 2 layers. VGG19 and exception are larger, deeper networks, which allow image recognition, object recognition in images. And we wanted to know how will they all do based on these very simple stimuli. And the way we encoded it from a computer science perspective was like this. So here's, for instance, an angle, right? And the angle here is 77 degrees. So we map this value from 0 to 90 degrees. We have stimuli from 0 to 90 degrees. We map it between 0 and 1. So this one would be a regression value of 0.856. And then we just train the computer, train the networks, those 4 networks, and yeah, with like a bunch of these stimuli, and then test them on a bunch of the disjunct stimuli and see how they're doing and calculate the error. And we do that for these elementary perceptual tasks, these 9, and then we also do that for the pie charts, for instance. And here for the pie charts, we encode it a little different. So this is the stimuli we generate, and we put a little dot in the largest area, and then we say, okay, identify the dot to the machine, and then go counterclockwise and compare all these segments area-wise to this largest segment. You know? So in this case, the target regression vector would be 0.2, which is, this is 20% of this one, this is 14% of this one, this is 60% of this one, this is 80% of this one. So that's how we then also generate these pie charts and let the computer learn on that. So with that, we trained over 2,500 different networks. And yeah, that's a significant amount of time. I had access to this large GPU cluster with 48 different GPUs and ran it for, I'd say, 1 month straight to run all these experiments and these trainings. And then the funny thing was, during the rebuttal phase of this paper, there was a fire in the data center, actually. I think everything went well, so that's why I'm laughing about it. But yeah, there was this fire, and then in the acknowledgements in this paper, we say like, hey, we promise that the training of the 2,500 networks did not cause this fire in our data center, you know, because yeah, we use so much computer and let the GPUs run at high temperature. So based on these 2,500 trained artificial neural networks, we were able to get some insights on how the machines perceive these elementary perceptual tasks and also the first steps of these charts. So let's look at this one. This one is position common scale. And the computer gets images like this, where there's a scale which is fixed, and then this dot just moves around. And then the computer is supposed to estimate the position of this dot on this line. You know? So that's very similar experiment to what Cleveland and McGill did with humans. So here we noticed the following. So first we took like the original Cleveland and McGill value, and this is a log scale, you know, the error. Here's no error, and it goes like to very over 50% wrong. Humans on all these perceptual tasks do like around 10% error, right? So then we noticed that the machine, especially VGG19, can do it so much better than humans, you know. And we added all kinds of things like extra noise, that the computer can't just memorize the pixels and so on. So I think it's a fair experiment. We noticed that all convolutional feature encoders, the networks here, you know, are doing better than the humans, while the MLP without any convolutions does a little bit worse than the human on average. You know. So here we have also, we trained VGG from scratch. We use pre-trained VGG. This is exception, pre-trained exception, and this is the Linet. You know. So all of these networks do much lower error when estimating this position common scale than the human or the network without any convolutional filters. Same for angle. Human around 10%, networks doing very, very well. Length the same, human, network. All right, VGG19 was actually the best network in all our experiments. Now, then we noticed the following. And here's something which I showed at the very beginning of this presentation. If we train the computer to measure the lines like this, you know, if we have thin lines, just vary the length, you know, the computer has no problem to learn to measure this length and does it with very, very high accuracy, you know. But now, if we move the line around, you know, like this, and I guess he is also a little bit moved around, but the more you move around, the more problematic it gets. Or if you make the line thicker, it does not work anymore. You know, the computer is not able to generalize the learned knowledge from the simple stimuli across these minimal variations. You know? So it's not possible. It only works if you add all these variations also as part of the training data. So this experiment is very important because it shows that even on very simple images, the computer is unable to generalize. And this is just such a milestone. We're so far away from a general artificial intelligence. And these simple experiments just show that. So you need to include all the variations to have the computer understand if you change something up. Now, for the pie charts and for the bar charts, we got the following results. Here's human between 2 and 10%. The VG a little bit better, not much difference. But what I found very interesting is that both human and artificial neural networks do better on the bar charts than on the pie charts. And that's something which is a common thought in the visualization community, where people say, okay, pie charts are not good for consumption of the values. It's harder to understand than the bar charts. You know, maybe think of that if you want to plot something. The visualization community thinks the bar charts are much easier to read. And we discovered that the machines seem to think the same, you know. And this is all the other networks. You see this trend here. For bar charts, they were more able to understand the encoded values than for the pie charts. All right, so this was presented 2018 in Berlin at the Visualization Conference. Now, Cleveland and McGill also did other experiments. Rather than single stimuli, single angles and so on, they now have stimuli where they have multiple angles and you compare them all in regards to each other, or multiple direction measurements, multiple areas, multiple volumes, multiple curvature values, and so on. So what we do now is we identify each of these stimuli, and this is ongoing research right now. We identify each one and then process them separately. It's like a similar concept like human selective cognition. You know, identify first, divide and conquer, and then process the stuff. And and then it works really well. We can identify each angle and measure it. But if we feed in the whole image at once, there are too many parameters, and VGG19, even trained from scratch, even trained with tons of data, is not able to do that very well. So we first need to identify, cut out each angle. We do that with mask RCNN. It's an instant segmentation network, and then we process it with the regression networks I trained from the old work. And this is a project where my PhD students, Jesse and Aswin, are working on right now. And for this day, we create a benchmark platform where we want to have standardized data sets where other researchers can test their algorithms, upload their results, and then get a leaderboard kind of situation. At the same time, I'm very interested in how the networks process these stimuli, and you can get those feature maps, right, to see which filters in which layer detects what. And also you can look at, let's say, attention, you know, like with those network dissection tools from researchers at MIT, for instance. And I have an undergrad student, but I'm looking for other people working on this. I'm very interested, where does the machine look if it identifies a pie chart, for instance? All right, so now maybe you identified the theme already. So I said there's this gap between natural and artificial intelligence, but what really works is when we combine the two. You know, I think that's a common theme, and many people are fans of that. The AI will not replace the human. What we need are systems which guide humans to be more efficient. You know, it's a combination of AI and human. And one of these systems we presented at Eurovis earlier this year, actually Best Paper Award. It's called Peaks. You know, my colleague Fritz took the lead there. and it's a system for genomic sequences and pattern search in genomic sequences. The user can identify a pattern and then, or like a peak, and then it finds similar patterns across multiple other sequences. You know, it's a web-based interactive region of interest selection. Very nice work, and Fritz is just so good with the visuals always, you know, with the graphics, and that's why we got the best paper award. Very proud of that. Another thing is, current work is the intelligent annotation framework. And I said before, the guided proofreading system, remember when the users gets yes, no selection and so on, at one point the classifier stalls, the user stalls, we don't get better, you know. And what we want to do is use an intelligent annotation framework to tune these automatic classifiers. And this is in the context of breast cancer detection. So we'll have two artificial neural networks. And one is the original classifier, which gives the first initial automatic segmentation. And then there's a second network. And they both work together for quality control. The discriminator, the second network, has only one job: to see whether an annotation, a segmentation, comes from an algorithm or from an expert, you know. And if the discriminator is not able to tell where the annotation comes from, that's perfect, because this means the classifier is as good as an expert. But in most cases, it would say, okay, this comes definitely from a machine. It will identify an error pattern in the classification, and then we can use some active learning and relabel and re-add as training data, have the human in the loop to fix this classifier, to fine-tune this classifier. And we actually, my research lab just got a significant amount of funding from the Massachusetts Life Sciences Center, Bits to Bytes program, to pursue this research in terms of breast cancer detection. We're working together with a company called Deep Health. Some of you might know Greg Zorensen is a co-founder. We're like working on this together now. And Bill Lauder, also very, there's another co-founder, David Cox as well. They are great guys, and I'm very happy that we got this funding together. And we will fine-tune the automatic breast cancer detection algorithm. And we already did some experiments on the guided proofreading with the setup. And in fact, this two-network setup was able, in just 3 cycles, was able to increase in the toy example, in a simulated experiment, increase the accuracy from 0.6 to, I think it was over 0.9. So under 0.6 to over 0.9, just for this two-network setup. So I always say, okay, 2 pairs of eyes see better than 1, and that's exactly what we simulate here. It's inspired by generative adversarial networks, but without the combined loss. All right. Well, I showed you a bunch of projects here. I know it was a lot, but that was my intention. I just wanted to give you a broad overview of all the work we're doing. And there's more. I could add more to the mix, but I know it was a lot to decompress here. So please reach out if you have questions. And I'll share, of course, the slides. It's a web link. You can just go to slides.com/Haehn/FNNDSC to actually see these slides. I'll put them in the chat right now, but we can, I'll send it also to Rudolph or Alan later. And now, this was my research, you know. And now I want to talk a little bit about the teaching, which you do also as a faculty member on the tenure track. And I'll do it quick, I only have limited time. But I'm teaching right now 2 courses, 1 each semester. So in the fall, I always teach computer graphics. And in the spring, I teach software engineering. And both are open education courses. You see the material online. In the spring, last spring, we did this, CS for 10 software engineering. And their slogan is real projects, real impact, real skills. And for the skills, I put a $sign because I like to include new computer science topics such as deep learning, Docker containers, Sizen bindings into this, you know. And what we do is we work together with different organizations. And one of the organizations was actually you and Rudolph with the FNNDSC. And he did a great job in mentoring the student project. And I'm not sure if they did something useful, but I hope they did something useful. Rudolph said so, at least, you know. So those are, yeah. In general, I would say every student group made something useful or like learned something, and that was very, very good. And in the fall, I teach computer graphics. I did that last fall. I want to show this video. It's one of the assignments from mid-course. So we do some crowd simulations, all in WebGL, of course, running in the browser. Some students add like fancy effects. Some students add like Tom Cruise into the mix. Some students add those like bump maps. So very, very cool work. It makes me always very proud to see how the students do this stuff. What I said at the summary of this presentation, we do it at different scales. And indeed, we start with XDK, you know, because XDK is still the most easy to use WebGL framework there is. You can just write 6 lines of JavaScript code to render something, render a mesh, render a cube, do some experiments. Then we learned 3JS, which is the most popular WebGL framework. Amy, your framework, the new medical imaging tool, is based on 3JS. And then we also do some stuff with native or like, WebGL without any framework, you know. So this is the complexity goes up, right? It's harder to use the WebGL. Well, XDK is easy. And then you have, of course, limited functionality versus more freedom. People do like pixel art like this. I love that. That's assignment number 2. Yeah, we see here you see the increasing complexity of the different classes we use, the different method calls. What we also do is a video game. I don't know why it's bouncing. Oops, that's a little annoying. So they're also, that's actually in WebGL, they designed this video game, you know, and the students, this student added the shooting feature. I don't know why it's bouncing, sorry for that. But yeah, very cool to see the variety of different student submissions. Here's another student, you know, added like a different background and so on. So then we do a field trip. Unfortunately, this year it's not going to be possible. We were virtual field trip this year, but we did last one. And then we do some experiments with this 360 camera, and we render. This is now rendered in the web browser. We create this. It's called like a sphere box. You know, we render the 360 video inside this box, and like do some experiments like this. And what you might like is that I had a guest speaker, and I will have him again this year. You know, this fall, Dan Ginsberg. So fantastic. You know, he also was at the FNNDSC before I joined even, you know, great guy, very accomplished computer graphics researcher. And yeah, he always gives great presentations in my course. So I'm looking forward to have him again this year. Next year, probably, most likely, in addition to software engineering, I'll teach a biomedical imaging course. And then I'm also looking for guest speakers. So yeah, I'll also reach out to you all. Maybe you can help me with that. And with that, I'll say thank you very much. You know, you can give me feedback here, anonymous feedback on the Rate My Talk. I do that after every lecture. And yeah, let me know if you have questions.
Great, thanks, Dan. That, as always, is fascinating and very creative, out of the box. And it's always wonderful to hear you talk. And I can bet that you're a real inspiration for your students too. There's so many things and questions that I have, but first I'll open the floor to see if anybody else has any questions that they want to ask.
I'm going to quickly jump in, Rudolph. Dan, that was a great talk. Thanks. It was amazing to see your work and what you're doing. Yeah, and we're certainly happy from our side to keep working together and collaborating. It's really amazing stuff that you're doing.
Excellent.
Yeah, no, it's a lot of fun. I'm gonna quickly start with one kind of larger, more philosophical question, just to get your feeling about a particular idea. So in the beginning of your talk, you compared, like, you showed the robot doing soccer against a human. You know, and that's a very compelling image, right? It's showing the difference, you know, that huge gap between these artificial networks and biological ones. But, you know, I'd argue almost you don't have to show a human, you can even show an insect, right? To show the scale difference, how you know, majestic or how powerful an insect is able to move and solve these, you know, do the information processing with so many fewer neurons, right? There's definitely some organizing principle at play here that we just simply don't get. And I have no idea when or if that will be better understood. But I wanted to ask you, the philosophical question was about the words that you use in talking about this field, which we all do, right? I mean, we all use these words like learn and train and understand as AI kind of practitioners. But I'm wondering lately if those words are actually bad ones to use in this field, because they create this idea, right, of something magic in a neural network or even in AI. There's no understanding at play here. When we use words like train, we think of a biological thing a little bit, like a dog who trains to sit or whatever. You know, if we use these words, we can talk about streams, I guess, that flow down a mountain, and we can say, oh, the stream is learning how to flow down the mountain. That's about the kind of intelligence or understanding or that exists in my mind in these neural networks, because so that's why I'm very careful these days when I talk about these tools to talk about them as an optimization problem, or to talk about a neural network as just finding a boundary in a hyperspace, because that kind of removes some of the mystique about it. And I think it's important to remove the mystique about these tools so that people first don't overhype them and think that they can do everything. But I'm just wondering what your thoughts are about this, about the words we use to describe this kind of technology.
So I totally agree with you, Rudolph. We are far away from anything even close to an insect. That's also, I think, some MIT research, some famous MIT AI research said we have not, we're far away from having anything close to an insect. Robots taking over the world will not happen in the near future, you know. So that's very important. But I think the field of AI has so many different aspects, from ethics over to technological challenges and so on. And what I really think, and many people think that, is that if we understand the brain a little better, we can model it better, and then we maybe can finally create something like this intelligence. And my student Yahia, who works on this mitochondria alignment, actually just went to summer school at MIT to the Dicalo lab, and they do this thing called brain score, right, where they try to create this core net, core object recognition network, which mimics the visual ventral stream, and then they compare it against measurements in macaques and monkeys, and this network actually, compared to all other existing deep learning networks, works better, or like mimics better how the ventral visual stream works in these monkeys, you know. And I know Yahia, and I'm excited to hear his latest research, what he did over the summer, works on something in addition to that, another part of the biology mapped into a neural network. So I don't know the details yet, but I think that is something we need to do in the future, you know, look more towards the biology. That's why I'm such a big fan of this connectomics, where we look at the cell connectivity at this scale, you know, to understand the networks. And also the same lines, I'm interested in this open worm project, right? C. elegans, we have the full connectome. Now we have the simulated open worm where we can, with some reinforcement learning, some agents, we can simulate or play around with this connectivity graph. And that's something I want to look into the near future. So I agree, we're very much at the beginning with all of this. You know, there is no real artificial intelligence yet. You know, it's all like statistics and so on. However, it is built off some neuroscientific insights, right? So we know how to model the individual neurons, and that's played into the design of the artificial neural networks and so on. And of course, you know me, I like to sell well, you know, I always like to show pretty pictures and hyped up words, because people in general buy that. I think it's important to communicate science at all levels for people who never, who are not experts, right? So that should understand what you're doing. So it's important to keep it simple and approachable. And I think these hyped up words actually help with that, you know, so it helps us to go more towards the sci-fi, more the attraction of the masses. And I like that approach a lot, and it certainly helps with some funding. As I just said, we got this intelligent annotation framework funded, you know, by like significant amount of money. So yeah, that's just how it goes. But I agree, yeah.
Yeah, that's good. That's really good.
And another concept that you were saying that I thought was really important too is the lack of generalizability of most of these networks, and coupled with your desire to understand how they are working and how they're recognizing things. And I think that, you know, is really important when we think about taking these networks into the medical field, because the network only learns what you have given it. So the whole generalizability question is of course, big concern to the medical community, because we don't know, you know, how, because we are so good at generalizing, it's hard to know if we've got enough data to do, you know, an accurate classification in a novel, you know, data set. So understanding better what the networks are keen on is, I think, really important to leverage those networks well and understand their limitations. What are your thoughts on that?
Yeah, so I still think, even if some people claim we don't even know how these deep learning networks really work, you know, there's always this level of uncertainty. The initialization, how you initialize the weights, plays an important role, you know. For these, looking at the machine graphical perception, we ran each training at least 4 times, up to 10 times, you know, just to get like a feeling if this is reproducible and so on. So there are many uncertainties just even from the simple models we have right now. So I would not trust the computer much, you know. So that's why I say we need this combination of human and AI, and especially in the medical field. Yeah, this project, the Intelligent Annotation Framework, working together with this company Deep Health, they have excellent results where they do breast cancer detection and compare it against expert radiologists, and there were high-profile radiology researchers involved. And the studies show that the algorithm does better than them on average, right? But then you also have yeah. So I think that the numbers don't lie, of course, you know, they did a good experiment, solid science, but still there's nothing which can really replicate in the computer what the real world is right now and the real world challenges, and especially in the medical field. So yes, caution is definitely what we need.
I'm curious, for that breast cancer example, if you just train it against the readers, the radiologists, they're not perfect, so they do make mistakes. Did you have actually outcome data in terms of whether there was or was not true cancer, you know, in the biopsy? Because I think really what you want to do is get that outcome data and train the network on not that, and actually go back then and train the radiologist to get better at what they're doing if you've trained it.
Yes, exactly. Yes. So of course, like we have large amounts of data, and as part of this grant, we want to release the world's largest annotated mammography data set. And yes, we have biopsy data for, I think it's 70,000 studies, and that's important, right? That's the real ground truth. We need that, you know.
I have another question, and I'm going to ask another question second. Just going back to other things, the Draco. Does that also, in the data that's being displayed, is tracked with that all the parameters, like in terms of, you know, the mean diffusivity, FA, you know, all those different parameters that we calculate? Is that all embedded in those files?
Yes, if it's embedded in the file, it stays embedded. Everything. So these mean FA and the others, it's called like per fiber and per vertex scalars you can attach, or property in Draktography it's called, I think, scalars and properties. And Draktography itself has this limitation of 10 of each or something. But yeah, in Draco you have flexible number, anything gets encoded and compressed. That said, what I didn't mention explicitly is Draco is a lossy format, you know, so there's some information lost. But we did in the Mikai paper some interesting experiments where we replicated published Draktography studies. So what we did, we took the data, compressed it, rerun the analysis, and we had the same results as the original published study on uncompressed data. And that's how we say, all right, the lossy compression is still so good, you know, based on the voxel resolution that you can skip these information, this information. And we were talking about 10 to the -5 or something of values, depending, of course, on the range of what you're mapping to the different fibers, you know. But yes, all this data is included in the files.
Cool. Any other questions? So I give other people a chance. Now, Daniel, maybe I should let you go. We're kind of running over time because there's just so much. I could keep asking questions for a couple hours more. So I would love to follow up with you and strategize and how we can develop more collaboration, because there's everything from, you know, some of the segmentation analysis that might be nicely pulled over into some of the image segmentation, where there still are challenges in terms of both, you know, combining regions that shouldn't be combined, and segment has got the same dual kind of issues. So there's, again, your work is just phenomenal, and so many potential intersections with what's happening on the medical front that it would be great to explore the collaborations more. And it would be wonderful to have an excuse to work with you again too.
Yes, I would love that. Absolutely. You know, I never lost touch with Rudolph, and that is so great too. And yes, I'm open to any collaboration. Yeah, I love the FNNDSC. I'm happy that you didn't change the name. You know, I remember the discussions back in the day, and I think it's going great.